You know, back in biology class, we all learned that simple story. DNA makes RNA, and RNA makes protein, the central dogma. But honestly, that's like having the architect's blueprint for a massive skyscraper, but having no idea how the electricity, the plumbing, or the elevators are supposed to work. Today, we're going to pull back the curtain on the hidden operating system that actually brings that blueprint to life.
Okay, so this is the huge shift in thinking we're talking about. On one side, you've got the blueprint, we all know. That's the static information. But on the other side, that's the operating system. These are Post-Translational Modifications, or PTMs for short, and they are the dynamic, real-time instructions that turn a list of parts into a living, breathing, functioning machine. And that leads to a really big question, right? We have poured billions of dollars into mapping the human genome, the blueprint. But what good is a perfect blueprint if the software running it is completely bug-ridden? What happens when this biological operating system crashes? Well, the answer, as we're about to see, is disease.
So to really get the connection to disease, we first need to understand what these PTMs actually are. The best way to think about it is to imagine the cell has this master control panel, just covered in switches, dials, and tags that control absolutely everything a protein does. That's the world of PTMs.
So here's the core idea. After a protein is built, fresh off the cellular assembly line, it's pretty much a blank slate. PTMs are the little chemical edits or post-it notes that get stuck on it, giving it its marching orders. Turn on, turn off, go over there, or, hey, it's time to self-destruct. They are the real directors of the whole cellular show.
And this isn't some tiny, obscure biological process. We are talking about a system of absolutely immense scale. Think about this. Over 90%, 90, of all the proteins in your body are chemically tweaked by PTMs. This is the rule, not the exception. The operating system is running on pretty much everything.
Now, scientists have found something like 300 different types of these edits, but really 5 of them are the superstars that dominate the story of health and disease. You've got phosphorylation, which is basically the cell's main on-off switch. Then there's ubiquitination, that's the delete tag that marks a protein for the garbage disposal. Acetylation works like a volume knob for our genes. Glycosylation is like a cellular ID card telling cells who's who. And finally, sumylation is a precision tuner, just making tiny adjustments. Each one is a totally critical tool in the cell's toolbox.
To really see how powerful this whole system is, let's just zoom in on one of these, the most common, and you could argue the most important PTM of them all. That simple little on-off switch, phosphorylation.
So how does this on-off switch actually work? Well, imagine a growth signal hits the outside of a cell. This wakes up an enzyme inside called a kinase. And the kinase has one job. It grabs a tiny phosphate molecule and slaps it onto a target protein. Boom! Just like that, the switch is flipped to on, and that triggers a whole cascade of commands telling the cell to grow. But, and this is the crucial part, if that switch gets stuck in the on position, you get the uncontrolled growth we see in things like cancer. That's why there's a whole other team of enzymes, called phosphatases, whose only job is to go around and flip those switches back off. It's this constant dynamic balancing act.
And that delicate balance, it is everything. The system is so elegant and absolutely essential for our health. So you can imagine, when it breaks, the consequences are, well, they're devastating.
And look at this. You can draw a straight line from these PTM failures to some of the diseases we fear the most. In a ton of cancers, the root problem is what we call hyperphosphorylation. Those growth switches are just permanently stuck on. In Alzheimer's, a protein called tau gets overloaded with those same phosphate switches, causing it to clump up into toxic tangles that kill brain cells. And in Parkinson's, it's often a failure of that delete tag system, ubiquitination. The cell's garbage disposal just breaks down, and all this toxic protein junk builds up.
And this really gets to the heart of it. In so many of these diseases, the genetic blueprint, the DNA itself, is perfectly fine. The problem isn't the hardware, it's the software. The PTM control layer is what's broken, and it's sending all the wrong signals throughout the cell.
Okay, so if the problem is a broken control panel, the solution seems pretty obvious, right? We need to figure out how to fix it, how to rewire it, and that is exactly what a huge part of modern drug discovery is all about.
Remember that stuck-on switch we talked about in cancer? Well, an entire class of drugs was designed specifically to fix that one problem. They're called kinase inhibitors, and their whole job is to find that rogue kinase enzyme and physically block it from flipping any more switches. It basically jams the switch in the off position. And this is not just a theory in a lab. It's a medical revolution that's been happening for the last 2 decades. It really kicked off in 2001 with a drug called imatinib, which proved this whole approach could be life-changing. Since then, and especially in just the last 5 years, we have seen an absolute explosion of these drugs, leading to nearly 100 approved medicines that all work by targeting this PTM control layer.
The impact on patients has been incredible. And the financial side of it, it's just staggering. Look at this chart. Back in 2001, this was a $0 market. Today, this single class of PTM targeting drugs is a cornerstone of modern medicine, bringing in over $30 billion a year.
But of course, science never stands still. Just blocking a broken switch is powerful, no doubt. But what if you could do something even more clever? What if you could hijack the cell's own machinery and turn it against the disease itself?
This slide just lays it out perfectly. The old way, inhibiting, is basically a roadblock. The new way, using these brilliant molecules called PROTACs, is more like a targeted drone strike. A PROTAC is like a molecular matchmaker. With one arm, it grabs onto the bad protein you want to get rid of. With its other arm, it grabs the cell's own delete tag machinery. It literally forces the cell to mark its own enemy for complete destruction. You're not just blocking the problem anymore, you are eliminating it.
So this evolution in thinking, from simply blocking a system to actively hijacking it, it really points to a much bigger idea. We're moving beyond just hitting single targets and toward reprogramming entire cellular systems.
So why have these PTMs become the absolute hub of drug discovery? Well, it really boils down to 4 huge advantages. First, target expansion. They give us hundreds of new targets we can go after. Second, functional precision. You can fine-tune a system instead of just using a sledgehammer to turn it off. Third, they're druggable. These enzymes have these perfect little pockets that drugs are designed to fit right into. And finally, the network effect. If you hit one master switch, you can reprogram an entire disease network all at once.
And the results, they're undeniable. Just look at oncology, one of the toughest areas in all of medicine. Since 2010, drugs that target the PTM operating system account for a massive 40% of all new small molecule cancer drug approvals. This strategy isn't just promising, it's become dominant.
So where is all this headed? Well, the next great frontier is combining our deep knowledge of PTMs with the incredible power of AI. Just imagine being able to map out a single patient's unique PTM profile to find their specific software bugs and then design a drug that precisely fixes their individual problem. That is the future. That is true personalized medicine.
And that brings us right back to where we started, doesn't it? The central dogma gave us the blueprint, which was a monumental achievement for science. But it's the PTMs that are the operating system, the living, dynamic software that ultimately decides whether that blueprint builds a healthy body or one that's ravaged by disease.
So I'll leave you with this final thought. For the last century, medicine has mostly been about finding a magic bullet to block one bad thing. But what if the future is completely different? What if it's about all of us becoming biological systems engineers, learning how to access the cell's operating system and literally rewriting the code to reprogram disease back into health?